Good day, everyone. This is Dicot Medical, and today we're going to be talking about muscle histology. In our last two videos, we spoke about the cell, the components of the cell, and the relation of the cell to the natural human body. Today, we're going to be talking on muscle histology, but first, we're going to deal with muscle as an organ. If you remember our steps from cell to tissue to organ to system. We're going to be treating muscle as an organ. We'll talk about the functions of the muscle, the properties of the muscle, and all the other things that make muscle an organ. Now, if the muscle is an organ, what system is it connected to? The muscle is connected to, or is a part of, the musculoskeletal system, which is a system that is the combination of muscle and the skeleton together. We're going to be treating different types of muscle, and we're going to treat the cell types under these types of muscle. Stay tuned as we learn about muscle today. The first thing we're going to be doing today, like I explained, is we're going to be talking about the muscle as a whole. So this is a drawing or a diagram of the muscle. It's just a rough sketch of the muscle. And here, the diagram in blue, the one in blue is the muscle, and the one in black is the bone. All we know about muscles, biceps, triceps, we know that muscles attach to bone to perform movement. Anatomically, muscles are organs because they are supplied by some particular structures, which I will be mentioning now. So the first structure is a nerve. Now, a nerve is a vessel that has no lumen. A nerve is a vessel that has no lumen that functions to transmit electrical impulses. It functions to send information from the brain or the spinal cord to an organ, and in this case, the organ is the muscle. It is because of the nerve impulses from the brain that the muscle can actually perform its major function, which is movement, like this. Okay. The brain is what instructs the muscle to do whatever it does. Secondly, we have an artery. So an artery is a blood vessel. There are two blood vessels, arteries and veins. But then an artery is a blood vessel which carries highly oxygenated blood. That is a blood that has high content of oxygen. And a vein is also a blood vessel, but a vein contains low oxygen, which means it has high concentration of deoxygenated blood. High concentration of deoxygenated blood. So arteries and veins actually communicate, but they communicate via capillaries. We won't go deep into that today, but what I need you to know about this is that where an artery is coming from the heart to an organ, bringing oxygenated blood to the organ. When the organ receives oxygenated blood, when the muscle receives oxygenated blood, it makes use of it. It passes out some end products, such as CO2. Now, those end products will pass into the vein, which is taken back to the heart. Arteries come from the heart, veins go to the heart. Again, what makes our muscle an organ? The nerve, the artery, the vein, and it also has some specific functions. Great. So we're going to talk about the functions of muscle now. There is an acronym for the functions of muscle, and you need to use it for this particular purpose only. So the functions of muscle acronym is MHB. For remembering or for remembrance purposes only, you can keep it as my hot body. My hot body. So the M stands for movement. The H stands for heat production. Hot, my hot body. And the B stands for body posture maintenance. Okay. As I said earlier, the major function of muscle is for movement, moving structures within the body or moving structures outside the body via the body. This is a pen. This is a marker, a pen marker. I am able to write with this because of the movement of my muscles. There are intricate muscles involved in writing. Those are fine muscles or delicate muscles. But then there are also coarse muscles involved in my writing. Internally also, viscera is surrounded by muscles, and even our blood vessels are surrounded by muscles. But all muscles are not of the same type. We will go into that later. What I need you to grab right now is that movement is the major function of muscle, and it does this via contraction, heat production. Now, muscles contain, or the muscles make up 40% of total body weight. As discussed earlier, we have over 100 trillion cells in our body. That means our muscles alone contain about 40% of those cells, almost half of our entire body weight. As a result of respiration in cells to produce energy, heat is released. A high concentration of cells in muscle means there's going to be a high concentration of heat released in the muscle. This is also why, or this can be attributed to strenuous exercise. In strenuous exercise, running or jogging for a particular amount of time, you notice that you are sweating, you're beginning to sweat. This is majorly, not only, but majorly due to the fact that your muscles are producing so much heat. The cells of your muscles are producing so much heat that the body needs to cool itself down. Also, or in relation to this, muscles also act as a form of insulation for the body in cold temperatures or in cold areas, cold seasons or weather. Lastly, body posture or maintenance. As you're sitting down right now, or you're standing, you're walking, you may feel like you are relaxed. To you, you are relaxed, but there are some muscles in play that help to keep your spinal cord upright. That is, if you are sitting straight right now. And even if you're not sitting straight, if you are bending like a banana, there are some muscles that are also involved, holding you upright. For example, your head, whether you are sitting straight or you're sitting like a banana, your head is held upright at the atlanto-occipital joint by some specific muscles. You can go and find them out later. All I'm trying to say is that the major function of muscle is movement, a byproduct of muscle cells, which is also a huge function of muscles because of the weight or the mass of the cells attached or the cells in muscle, is heat production and body posture maintenance. When you're standing, when you're sitting, when you're laying down, muscles are involved. Now we're going to talk about the properties of muscle. My hot body is an acronym for the functions of muscle. We're going to talk about the properties of muscle, and the acronym for that is ICE. ICE with an extra E. If you want, your acronym instead of ICE can be extensible ICE, even though ICE is not extensible, but that's if you want. So the I in ICE actually stands for irritability. Let me write it straight out. Irritability. What does irritability mean? Irritability means that muscle can actually react to a certain stimuli, whatever stimuli it is. But there is a particular stimuli related to the function of muscle. But irritability means that when a stimuli is attached or when a stimuli is applied to muscle, there's going to be a reaction, and that reaction is called excitation. C stands for contraction or contractility. Okay. I said the reaction of muscle to a stimuli is contraction. or is excitation. Now that excitation it reveals itself via contraction. Contraction is the shortening of muscle in relation to a nerve impulse, in relation to messages or information flowing from the brain to a particular muscle. Contraction, lengthening, contraction, shortening. Next, we have extensibility. Extensibility means that after contraction takes place, the muscle can actually stretch out. Like after contraction takes place, when contraction takes place, extensibility means that when contraction of a particular muscle takes place, there is going to be an extension of another muscle adjacent to it. Extension to be stretched out. Extension or extensibility also means that after a muscle undergoes contraction, it relaxes back or it lengthens. That's extensibility. Lastly, we have elasticity. We can remember elasticity from our physics, but in case we don't, elasticity means, or elasticity means that after a particular material is stretched, it has the ability to return back to its normal position. In relation to muscle, elasticity means that after it has been stretched via contraction or via extensibility, it can return back to its appropriate size. This is important because in extensibility, there are some situations where muscle is stretched more than it can handle or stretched farther than its original length, which is in action potential terms called hyperrepolarization. This will be discussed in another video. But for now, extensibility means lengthening. Elasticity means the ability to return back to the normal length of the particular muscle. Just a very quick recap. We've spoken about muscle as an organ because it is supplied by a nerve, an artery, and a vein. We've spoken about its functions as an organ, its properties as an organ. Now we're going to talk about the histology of muscle. That's muscle histology, the main thing in this video. For some of you who may know or some who don't know, histology is the study of cell and tissues. The study of cell and tissues. Muscle histology will be the study of the cell and tissues of muscle. Muscle is an organ. What then is the tissue of muscle? Muscle is an organ. What is the cell of muscle called? Those are some of the things, or those are the foundational things we are going to be dealing with in this video right now. It is important to note that in muscle histology, there are 3 types of muscle: skeletal muscle, smooth muscle, and cardiac muscle. Skeletal muscle has its name, or gets its name, from the fact that it is most of the time, about 80 to 90% of the time, attached to the skeleton, that is the bones. Skeletal muscle most of the time is attached to the skeleton. The diagram I showed earlier was an example of skeletal muscle. Smooth muscle. Smooth muscle is found in the viscera. The viscera, that is, it can be found in the gastrointestinal tract from our previous video. It can be found in the respiratory tract, even in the blood vessels we just discussed. Blood vessels, not nerve. Blood vessels we just discussed has smooth muscle. Lastly, cardiac muscle, from its name, heart muscle, muscle of the heart. It's a muscle type which is found only in physiological conditions in the heart. We've spoken about types of muscle briefly. We spoke about it briefly. We're going to go deeper into the types of muscle, starting with skeletal muscle. Even though these 3 types of muscles, skeletal, cardiac, and smooth, are all muscle types, there are some certain characteristics or properties that separate them from each other. I'm going to be starting with skeletal muscle, and I'll be using it as the basis because it is the most common, the most functional, and it is the one most of us can relate to. I have an abbreviation for the properties or the characteristics of muscles in general. This will help you to differentiate each muscle type. I'll be applying that mnemonic of the characteristics of muscles to skeletal muscle as we go deep into it now. The mnemonic is strong clinics. Strong clinics. Don't be alarmed, we are not going to be using all of the letters in the words, in these words, but we're going to be using some of them. Strong clinics, we're going to bring out S, then C, L from the clinics. We're going to bring out another S, then I, N, C, L, I, N, clinics. Then finally, we're going to have an R, then we are going to have double C's. The first S in strong clinics stands for shape. The C stands for control. L stands for location. The other S stands for striations. N stands for nuclei. I stands for intercalated disc or discs. R stands for regeneration. C, our second C, stands for contraction threshold. And our last C stands for connective tissue. These are the characteristics we are going to be using to differentiate each muscle type, or we are going to be using to describe each muscle type. The structure above me, this structure, is actually a diagram or a sketch of skeletal muscle. Remember, muscle is an organ. This skeletal muscle as an organ is actually made up of different bundles. This organ is made up of tissues, and then these tissues or bundles are called muscle fascicle. So let's start labeling. This big one is called our organ, our muscle. It's our muscle itself. Bracket organ. This one, it looks like it has been brought out. You can see these little bundles, these little things inside this organ. Now, these little things are actually called muscle fascicle. It's called muscle fascicle because it is a bundle of cells. And what is a bundle of cells, or what contains cells? Tissue. So this muscle fascicle here, muscle fascicle. This is our muscle fascicle. It's a bundle of cells, and it's going to be our tissue, or it is a tissue. So tissue. Remember, bundle of cells. Remember, organ, bundles, or it contains bundles of tissues. This here that has been brought out of our bundles of cells. If we have a bundle of cells and we bring, and that bundle has some things inside, and we bring one out, what's that going to be? That's going to be our cell. Okay, so this is muscle cell or muscle fiber or myocyte. What does that tell us? That tells us that the cells of muscle, skeletal muscle, smooth muscle, cardiac muscle, but right now we're focusing on skeletal muscle. The cells of skeletal muscle are actually called muscle cells, muscle fibers, or myocytes. So the first characteristic we are going to be dealing with, shape. What is the shape? As you can see, this is our cell, our muscle cell. What is the shape of our muscle cell? Our muscle cell is cylindrical in shape. So we're going to come here and write cylindrical. This is the shape of a cylinder, so our muscle cell is cylindrical. The second characteristic is going to be control. Remember we spoke about how muscles are able to cause movement, which is via the transmission of nerve impulses from a nerve. That is nerve, like the transfer of nerve impulses from a nerve, innervation. Our skeletal muscle receives its own nerve impulses through the somatic branch of the nervous system. The control of our skeletal system, of our skeletal muscle, is via somatic branch of the nervous system. The somatic branch of the nervous system controls voluntary movement. Why am I saying this? Why do I feel this is important? This is important because skeletal muscle, unlike smooth muscle and cardiac muscle, can actually be controlled voluntarily. It's voluntary movement. Muscles, this is voluntary movement. I choose to control it. You choose to sit down. It is voluntary movement. You are in control of most of your skeletal muscle movements. Most, because there are some skeletal muscles that are involved in reflex movement. But for the most part of it, about 95% of it, you can control your movement. So for control, we are going to write somatic nervous system. Remember, somatic nervous system and voluntary. The somatic nervous system transmits impulses from the central nervous system to enable voluntary movement. If I want to move my arm, I want to, but for the information to be passed, I want to, but for the information to be passed to my muscle, it's going to be via the somatic branch of the nervous system. Somatic, voluntary. The third characteristic is going to be location. Skeletal muscle, in its name, is mostly attached to parts of the skeleton, the different parts of the skeleton. Your skeleton, your skull, to your vertebrae, your clavicle, your scapula, your humerus, your radius, your ulna. Muscles are attached to these parts of the skeleton, and these muscles that are attached there are called skeletal muscle. So location is going to be skeleton. The next characteristic is striations. From what you can see, down to our muscle fiber, the entire muscle, the entire skeletal muscle, yes, it has these stripes, these lines, they are stripes, it has these stripes. Even down to our muscle cell, it has these stripes, and these stripes are striations. Striations or stripes. So are striations present in our skeletal muscle? From what you can see, yes. So present. Nuclei. So for this next characteristic, which is the nuclei of our muscle fiber, we're going to be focusing on this diagram. Elementary cell, as you all remember, has just one nucleus. But our skeletal muscle fiber, or the muscle fiber of our skeletal muscle, has multi or many nuclei. This is one nuclei, this is another nuclei, this is one nucleus, this is another nucleus, this is another nucleus. So this cell, just this cell alone, actually has 3 nuclei. This is just an example, an exemplar cell, already has 3 nuclei. Another thing about the nuclei of the muscle fiber in skeletal muscle is that it is peripherally located. In our elementary cell, the nuclei is centrally located, but in the muscle fiber of skeletal muscle, the nuclei is peripherally located. The next characteristic we're going to be talking about is intercalated disc. From what we can, what are intercalated discs? So intercalated disc is physically or histologically is actually a zigzag line present when there is a joining of two cells, when there is a branching or a joining of two cells. From what we can see here, our bundles of fibers, our muscle fasciculi, they are not joining together. And also from what we can see here, as minute as it is, our muscle fibers themselves are not joining together. So no, skeletal muscle does not have intercalated discs. No intercalated discs. And for nuclei, we said peripherally located. So peripherally located means that it is located around. This is the center of our muscle fiber. We cannot see any nucleus in the center, but we can see nuclei around. Peripherally located means the nuclei is around the outer part. The nuclei is around the outer part of the cell. That is, it is close to the plasma membrane of the cell. So peripherally located, and it is multi-nucleated. The next we're going to be discussing about is regeneration. Back to our muscle fiber or muscle cell, we can see some little brothers floating around the plasma membrane of our muscle fiber. Now, these things are actually called myosatellite cells or satellite cells. Let's go with myosatellite cells. So, myosatellite cells are actually mesenchymal cells. Scratch that. Myosatellite. In the embryology of skeletal muscle, in the embryological development of skeletal muscle, it is gotten from myoblasts. Skeletal muscle is gotten from myoblasts. So myoblasts are the cells from the mesoderm of our growing fetus or the growing embryo. Now these myoblasts actually come together and they fuse to form these long cylindrical muscle fibers. Some myoblasts are left around the muscle fiber or on the plasma membrane of the muscle fiber. This functions to aid regeneration, that is recreation or creation of new muscle fibers in the case of injury. As we can see here, for this muscle fiber, it has just 2 myosatellite cells. And even this is an exaggeration. Most muscle fibers have just 1 myosatellite cell. That means that although regeneration that is possible, that is creation or new creation after injury, is possible in skeletal muscle, it is very, very limited. So for regeneration, we are writing limited. The second C, or the second to the last characteristic we have, is contraction threshold. Now, all muscle types follow this threshold I'm about to talk about. They will follow this. So they follow the all or none law, meaning that if the stimulus, the muscle can do movements or muscle can undergo movement because of impulses from the nerve, impulses from a nerve. Now, if these impulses do not reach a particular threshold, no movement, no control, no excitation. You know, excitation is the fundamental part. Excitation is the beginning. If these nerve impulses do not reach a certain threshold, excitation does not occur, contraction does not occur. Contraction threshold. Again, if these nerve impulses do not reach a particular threshold, they do not reach a particular level, excitation does not occur, contraction of the muscle does not occur, and movement does not occur. So does skeletal muscle follow the all or none law? Yes, it does. And so does every other muscle type. The last characteristic we're going to be talking about under explanation of skeletal muscle would be connective tissue. Connective tissue is made up of extracellular matrix and cells. Extracellular matrix being fibers and ground substance, and then cells. The cell type we have here, cell type we can see here, is the muscle fiber. Connective tissue, the first place where we have connective tissue in the muscle as an organ. We actually have connective tissue wrapping around this muscle or this organ. This is a transverse section. If I cut this muscle, like this muscle itself, if I take a knife and I do cha, cha, and I bring it out, and I turn it towards you, if only we could flip it, if only this thing wasn't 2D, if only we could flip it, if I could turn it towards you and I take a slice of it, this is what it is going to show us. We can see that there's something surrounding, let's say this is the muscle itself. Now there's something surrounding it. Now that is connective tissue. And the name of this connective tissue surrounding the muscle itself is called epimysium. Epimysium. So epimysium is the connective tissue surrounding the muscle itself. Muscle as an organ. It is the connective tissue surrounding the organ, the muscle. We go deeper. Even in this muscle itself, we can see our muscle fasciculus, our muscle fasciculi, which is this. We can see it here, you know, this is what is here. But then we can still see some things here, some guys, some lines in here. What does that mean? More connective tissue. Now the name of the connective tissue that is in here, the connective tissue that is surrounding the muscle fasciculus or muscle fasciculi is called perimysium. 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 Again, epimysium is the connective tissue surrounding the muscle as an organ, the muscle itself, your biceps. Epimysium is the one surrounding that. But then the bundles of cells, that is the muscle fasciculi, the muscle fasciculus, is surrounded by a different type of, or a different kind, also connective tissue, but is surrounded by the perimysium. Now, if we are going to go microscopically, there is also a connective tissue type surrounding the muscle fiber or the muscle cell. Now, the name of the connective tissue surrounding the muscle fiber or the muscle cell, our main concern of today, is called endomysium. Endomysium. So, okay, let's just give this a double layer, and I'll explain why very soon. Pardon my diagram. Let's just give it a double layer. Okay, so it is endomysium. Endo, inside, endomysium. Endomysium is the connective tissue surrounding the muscle cell itself or the muscle fiber. These three types of connective tissue are more distinctly found in skeletal muscle. Okay, we're going to go into what should we do next? What do you guys think we should do next? Okay, how about we take each transverse section one by one? As I explained already, or as I explained earlier, this is a transverse section of this. That means if I cut this together and I bring out a slice and I flip that slice, bah, that is what is here. These things that we're seeing here, like these are fasciculi. Fasciculi. Muscle fasciculi. What do we say fasciculi are? Fasciculi are bundles of cells. So fasciculi are bundles of cells. These three dots you can see here. I hope you remember when I spoke about what makes an organ an organ. Apart from the functions and the properties of that organ, each organ or every organ is supplied by a nerve and two blood vessels. And those blood vessels are the artery and the vein. So that is what is represented by these three circles. Remember, a transverse section. If you have a pipe, you cut it, you cut a section of it, and you turn it towards yourself. You turn the hole, the part that has the hole towards yourself. This is what it will look like. So you can name this, so this nerve artery, and vein. Okay, great. Again, these things inside are bundles of cells. So, we're going to deal with our muscle fascicle now. If we take a piece, the transverse section of our muscle fascicle, this is what we're going to see. Remember, each muscle fascicle is a bundle of cells. If this is a muscle fascicle, what are these things inside? Cells. They are going to be cells. Which type of cells? Muscle fibers or muscle cells? This connective tissue. Which connective tissue did I say is going to surround our muscle fasciculus? If you said perimysium, you are very correct. So this connective tissue here is going to be perimysium. And this connective tissue inside is going to be endomysium. Very, very cool, very, very cool. We're going to talk about our cell itself now. Elementary cell, which we have spoken about already. If you haven't watched that video, please go and watch the video. Just click on my channel and you'll see part 1 and part 2. I care about you so much that I divided it into part 1 and part 2 so that you will understand properly. Now, from what was discussed or from what has been discussed already, cell has organelles. Each cell has organelles. Now, in muscle fibers, not just skeletal muscle, but since we are dealing with skeletal muscle, I will just say skeletal muscle. But in muscle fibers, we also have organelles, but the organelles, number 1, they differ in names, and number 2, these organelles differ in contrast, like they differ in components. Yep, the organelles differ in components. This outer part I said is going to be the connective tissue. It's going to be called, or it is called, endomysium. Now, the endomysium is actually attached to the plasma membrane of the muscle cell fiber. But in muscle, we don't call it plasma membrane. We call it sarcolemma. So this plasma membrane is sarcolemma, similar to plasmalemma. It's not that hard. You know it. The nuclei is still going to be called the same name, but this inside, I don't know if you can see it, it is the mitochondria. In a normal cell, it is mitochondria, but in muscle, we're going to call it sarcosome. I'm pronouncing this very well so that you will not go and do mistake. Sarcosome. Sarcosome. Now, another component or another thing we have in normal cells that we also have here is the endoplasmic reticulum. But in muscle cell, okay, we have a representation of smooth endoplasmic reticulum only. And that representation of smooth endoplasmic reticulum we have is actually called sarcoplasmic reticulum. So if we just put our reticulum here, let's put it here. Sarcoplasmic reticulum. So that's going to be called our sarcoplasmic reticulum. And then this space, the space where all the organelles are, in normal cell called cytoplasm, is going to be called sarcoplasm here. I'm sure you guys are smart. You know the trend. Sarco. We're adding sarco to most of these organelles. Okay, let's pop the big question. I'm sure you are wondering why you are seeing dots inside this cell. The functional unit of each muscle cell is actually a myofibril. You can call it the functional unit, or more relatedly, the contractile unit. It is actually a myofibril. Okay. We don't have a microscope with us right now, but don't worry. I've taken care of that. I've drawn our myofibril out for us. But let me tell you some things to note about our myofibril. So, our myofibril has some striations, okay? Remember, this is our muscle fiber, this is it. But then now, I brought something out. This is our myofibril. So, if we bring out our myofibril, it's going to also have striations, like you can see on this muscle fasciculus. But the striations are ordered and they are arranged. The striations are what? Ordered and arranged. And I'll be explaining why. Why does our myofibril, or why do myofibrils actually have arranged striations? It is because of this diagram right here. Our myofibrils have striations because of this diagram right here. Now, this diagram here represents the contractile unit of a muscle fiber. It is very important. You need to take note of this diagram if you want to learn muscle histology properly, which would also aid your knowledge in muscle physiology. Now, this diagram here, like I said, is the contractile unit, or displays the contractile unit of a muscle fiber. But let us begin our labeling process. Whenever you have a diagram, you have to know the parts of the diagram, what is important about that diagram, the functions of those parts, or the representations, what they mean of those parts. From this point to this point, from this point to this point is actually called something. It is called a sarcomere. Yes, sarcomere. So if we take this down and we take this down, from this point to this point, sarcomere. Now, what is the point to the point that we just finished deriving our sarcomere from? That point is actually called the Z line, Z band. I know it doesn't look like a straight line, but there are different types of lines. This is the zigzag line. So we're going to call it the Z line. Yes, the Z line. So this, so this line, we're going to call it the Z line, and this line too, the Z line. So what does that tell us? That tells us that the sarcomere is the distance between two Z lines. Two Z lines. The sarcomere is the distance between two Z lines. If you're looking at a myofibril, physically, microscopically, if you're looking at a myofibril from the longitudinal direction, this is transverse, this is longitudinal. So if we could, if only we could, bring out our myofibril, make it as big as this, and look at it on this longitudinal version, on this longitudinal section, we are going to notice striations. But in those striations, the striations, there are two different types of bands, or they have different depths. Let me explain. We have the light band and we have the dark band. If we look at this structure, everything that is black here is going to be our dark band. Okay, dark. I used black because black is supposed to be darker than blue. So this is our dark band, and this is going to be our light band. After this video, please ensure to research so that you physically see what I'm talking about, so that this will stick in your memory. Go to Google and search for microscopical view of the myofibril longitudinal section. Great. So this is our dark band. And our dark band, we cannot just write the old dark here, or I don't want to write the old dark here. So I'm going to give it, or I'm going to attach another name to it, which is its true name, the A band. The A band. A band means anisotropic in polarized light. Anisotropic in polarized light. And this part is going to be our I band. It is lighter. What did I just say? I said it is lighter. And I also said that on the myofibril, we will see dark bands and light bands. These dark bands and light bands are actually given their name because one is thicker and one is thinner. As you already know, or as you already may be able to tell, our dark bands are going to be thicker than our light bands. So our dark band can also be called our thick filament. And our light band is going to be called our thin filament. Okay, I'm going to be going deep into the functional unit or the contractile unit of our myofibril. Like I said earlier, the contractile unit of our myofibril is sarcomere. And sarcomere is the distance between two Z lines or two Z bands, whichever one you choose. Great. This line here that we're seeing, this line is called the M line. This horizontal part is actually what is the thick filament. This horizontal part is also actually what is the thin filament. This vertical line here is called the M line. And now the M line bisects the thick filament. It bisects the A band. The M line bisects the A band like this. It runs across the entire myofibril molecule. Where the M line is, where it bisects, there is a zone called the H zone. The significance of the H zone is this. Thick filaments are bound to the Z line or about to the Z line by titin. In the situation of contraction, or when contraction is about to take place, or when contraction is taking place, thin filaments slide against or slide on thick filaments. That means if this blue filament is sliding like this, that means there's going to be a shortening of the sarcomere. Now, as much as the sarcomere is going to shorten and this thin filament is going to go inside, there's going to be a small space in the middle where the thin filament does not overlap with the thick filament. That part where the thin filament does not overlap with the thick filament is what we call our H zone. Histologically, this is important because if you are opportune to see a slide of such high magnification in which you can see the sarcomere, you will be able to differentiate the thick bands from the thick bands. But in the thick band, you're going to observe a faded center. I mean, it's the center of that thick band is not going to be as darkly stained as around it. Why is that? Because that's the center where the thick filament, where the thin filament does not overlap with the thin filament. That is called the H zone. Okay, like I said, titin is what binds this thick filament to our Z line or our Z line. This is the overlapping zone I'm talking about, where the thin filament is sliding on the thick filament. Where the thin filament is sliding on the thick filament, both of them together. That's what I'm calling the overlapping zone. Good. Let's go deeper into our thick filament and our thin filament. So our thick filament, the comprising molecule of our thick filament is called myosin, and the comprising molecule of our thin filament is called actin or F-actin. So our myosin molecule, this is a rough sketch of our myosin molecule. Our myosin molecule has a head and a tail region, a head region and a tail region. The tail region consists of two heavy chains. The tail region consists of two heavy chains. At the head region, we have two pairs of light chains, two heavy chains and two pairs. These things that look like ears are two pairs of light chains, labeled light chain and this one heavy chain. Then the actin, F-actin molecule. It is the molecule of our thin filament. When you think about thin filament, your mind should go to F-actin. When you think about thick filament, the first thing that should come to your mind is myosin. In the F-actin molecule, It is made up of, firstly, 2 polypeptide chains in a helical manner. This is the diagram. I'm going to show you a better diagram, a better representation of what I'm about to say. This is it. This is going to be our F-actin molecule. This is this thing that I'm drawing out like this. I'm tracing with my finger. Is the polypeptide chains. Inside the polypeptide chains, or around the polypeptide chains, you can see that there's one line that is outlining it. That line is actually called tropomyosin. In the F-actin molecule, apart from G-actin is the circle small molecules that comprise the polypeptide chain. Apart from the G-actin, there are 2 other important protein components in the F-actin molecule. This rope that is holding the 2 polypeptide chains together is called tropomyosin. Tropomyosin gives structure and it holds them together. That's basically what it does. It holds the 2 polypeptide chains together. Troponin attaches to tropomyosin. And troponin is made up of 3 complexes, 3 important complexes. TNT, troponin N tropomyosin, as in troponin binding to tropomyosin. TNC, that is troponin binding to calcium, what is allowing troponin to bind to calcium. And TNI, troponin N interaction, that is what is allowing troponin to cause the contract. The troponin molecule is a very, very major component in our contraction. But just know that TNI, troponin NI, in troponin N interaction, is a major component in our contraction, in the contraction of a muscle fiber. Remember the H zone I spoke about. The H zone is the region where the thin filament does not successfully or does not at all overlap with the thick filament. This is our actin molecule, this is our myosin molecule. What exactly happens in the H zone is that since our myosin molecule has just a head region and a tail region, the head region of our myosin molecule is the only region that is able to bind to actin, to the G-actin of the F-actin molecule. What does that mean? That means our tail region does not bind at all. If we put 2 and 2 together, it is going to be the tail region of the myosin molecule that is making up or that makes up the H zone. Because in the H zone, thin and thick filament don't overlap. The tail region of the myosin is unable to bind to the actin molecule, therefore there is no overlapping, there is no joining, there is no binding, so that place is blank. In this, our actin molecule, we have troponin and we have tropomyosin. The G-actin strings are held together by the tropomyosin, which gives it structure, allows it to stay together. The troponin is bound to, this is the troponin is bound to the tropomyosin rope. The troponin has 3 complex subunits, or is a 3 complex subunit. It's 3. TNT, TNC, and TNI, which enables it to perform its function. Smooth muscle cells or fusiform cells, they don't have troponin and tropomyosin. Instead, what they have is calmodulin and MLCK. Calmodulin is what acts as the tropomyosin of smooth muscle cells, holding together the actin molecules, while MLCK is what acts as the troponin. MLCK stands for myosin light chain kinase. So, because of MLCK and calmodulin, smooth muscle cells is able to undergo contraction also. It's able to undergo contraction. The type of contraction in smooth muscle cell is rhythmic. And this contraction in smooth muscle cell, the contraction in cardiac cell, can be affected by hormones, some specific type of hormones. These types of hormones that can affect them are linked to whichever part of the system or which system it is. Cardiac muscle cell. The type of hormones that can affect the way it beats, the way it contracts, is going to be linked to the hormones of the cardiovascular system or hormones for the cardiovascular system. And for the smooth muscle cell, for whatever system, if it's vasoconstriction, hormones of the cardiovascular system, and if it has to do with the parasympathetic or sympathetic stimulation of the gastrointestinal tract, it will be the gastrointestinal system hormones. We are done with skeletal muscle, and now we are going to talk about cardiac muscle or cardiac type of muscle. Embryologically, cardiac muscle is derived from mesenchymal cells, and those mesenchymal cells align with the primitive heart tube. The heart tube is the primitive part. It's the primitive part of the adult heart. Your heart, when it starts forming, the beginning part, at the beginning it looks like a tube. That tube is called the heart tube. And then the mesenchymal cells align with the heart tube. They align with it, they align around the heart tube. And then they fuse together. Now, these cells fuse together. Those cells that fuse together, they form intercalated discs. And then they are able to communicate. One cell fuses with another cell. It is still an individual cell, but there is a fusion. It is able to communicate via junctional complexes. Junctional complexes, they are complexes that facilitate communication between cells. In explaining about cardiac muscle, we are going to be using the characteristics we listed out previously. Strong clinics. The first S stands for shape. The cardiac muscle does look cylindrical in shape, but then it also branches out. So it's cylindrical and branching out. This is our nucleus, which signifies that this is a cell. As much as this is a cell, muscle fiber, muscle cell, as cylindrical as it is, it branches out. As cylindrical as this is, it branches out. As cylindrical as this is, it branches out. So there are branches, so cylindrical and branching out. C stands for control. Although C stands for control. Although in skeletal muscle, we are able to control our movements most of the time, and skeletal muscle is supplied by somatic branch of the nervous system. In cardiac muscle, we do not control the beating of our hearts. The cardiac muscle is supplied by the autonomic part of the nervous system, and it is involuntary. The autonomic part of the nervous system is divided into the sympathetic and the parasympathetic part. In regards to cardiac muscle or cardiac muscle fiber, the parasympathetic part functions to decrease or reduce heart rate, while the sympathetic part functions to increase heart rate. What I'm trying to say right now is that dealing with the control of cardiac muscle or cardiac muscle cell or cardiac muscle fiber, it is involuntary, and it is done via the autonomic branch of the nervous system or of the central nervous system. So, autonomic, non or involuntary, let me write involuntary. The next characteristic is location. It's as implied in the name, or it is in the name. Cardiac muscle cell or cardiac muscle fiber is physiologically only found in the heart. Physiologically, meaning normal body function. When there is normalcy in the body, cardiac muscle or cardiac muscle cells are found solely in the heart. Location is the heart. Striation. Visibly, yes, as we can see, there are striations. These are intercalated discs, but then there are also striations in cardiac muscle. But they are not as eminent as the striation in the skeletal muscle. So just take this one, chop it. That's striation. So striations are present. Nuclei. Great. We said in skeletal muscle, our nuclei, our muscle fiber in skeletal muscle is multinucleated and it has peripherally located nuclei. Well, in cardiac muscle, it is centrally located and it is just as a single nuclei, so mononucleated. So mononucleated and centrally located. Intercalated discs. We have gotten to the owner of intercalated discs. So intercalated disc signifies the joining together of two cells, the interlocking or the interdigitation of two cells. In cardiac muscle, we have a lot of that. That is the only muscle where that's the only muscle fiber type or muscle cell type where we have interdigitation of two muscle cells, or where two muscle cells come together and join together. The intercalated disc is the representation of when those two cells come together. It is filled with many junctional complexes, cell junction, desmosomes, gap junction, which aid communication between those two cells that have fused together. For example, gap junctions aid in electrical communication, ion communication between the cells that have been fused together, or electrical impulse to the cell. So intercalated discs, yes, it has intercalated. So these are intercalated discs. Intercalated discs. Regeneration. In cardiac muscle, regeneration does not happen. Unfortunately, it does not happen. In the case of injury, that means when cardiac muscle cell or cardiac muscle fiber undergoes, or unfortunately is hit, takes a blow, and an injury occurs, there is no regeneration of the muscle fiber itself. What happens is that connective tissue fills up that space. And when connective tissue fills up that space, that's what causes scarring in the heart or artifacts in the heart, because there is no regeneration in cardiac muscle. So for regeneration, we have no. Contraction threshold, the same as skeletal muscle, all or none law. It has to reach a particular stimuli. If it doesn't reach that particular stimuli, then no excitation is going to happen, no contraction is going to happen, no movement is going to happen. Does it follow the contraction threshold? Yes, it does. Connective tissue. Great. The connective tissue of the cardiac muscle is similar but not exactly the same. Or yes, it is similar but it doesn't exactly have the same significance as in the skeletal muscle. Obviously, in cardiac muscle, we also have the endomysium. The endomysium is the connective tissue surrounding. This is we've gone down like this, and this is the cell. This is the cell itself of cardiac muscle. We have connective tissue surrounding it. This connective tissue, I hope you remember, is called endomysium. Now, surrounding the fasciculi of this cardiac muscle, fasciculi, bundle of cells. So surrounding the fasciculi, we also still have the perimysium. But what happens is that in the perimysium of the cardiac muscle, the perimysium contributes to the formation of the cardiac skeleton. Now, cardiac skeleton is made up of connective tissue. The cardiac skeleton, the structure, is made up of connective tissue, and it provides a framework, supportive framework, structural framework for the chambers and the valves of the heart. The cardiac skeleton provides structural framework for the chambers and the valves of the heart. And the cardiac skeleton has a great contribution from the perimysium. Great. And then outside the entire cardiac muscle, we have the epimysium. Now that epimysium is going to be continuous with the pericardial sac. The heart is inside a sac. The perimysium is going to be in contact, the epimysium is going to be in contact or continuous with the pericardial sac. Cool. This is our nucleus, this is our nucleus, this is our nucleus. This is the capillary. I told you now, blood vessel at microscopic level, it will look like that. That's our capillary. This intercalated discs. The contraction in cardiac muscle will be the contraction in cardiac muscle is similar and just about exactly the same as the contraction in skeletal muscle. But like I said, contraction of muscle, that's the physiology. The function of a muscle most of the time is movement. How does it perform movement via contraction? Physiology is the study of normal body functions. We're going to be dealing with the contraction of muscle, both skeletal muscle, cardiac muscle, mostly skeletal muscle, in another video. What differentiates, apart from these characteristics we've just mentioned, there are some other things that differentiate cardiac muscle from skeletal muscle. One very important thing to note is that cardiac muscle actually has a great amount or a larger amount of mitochondria than skeletal muscle. Cardiac muscle is made up, the cardiac muscle cell, the cell itself is made up of 40% mitochondria. Yes, what is muscle obsession with 40%? Cardiac muscle cell is made up of 40% of mitochondria. What does that mean? That is like very, that's a lot. That's much more than skeletal muscle. What does that mean? That means that cardiac muscle, as much as skeletal muscle needs energy, to perform our voluntary functions, cardiac muscle needs much more energy because your heart never stops beating. If the heart of an individual stops beating, that is death. When a person sleeps, the person is able to rest their voluntary muscles. They're able to rest their skeletal muscles. But even when you sleep, your heart beats. So it needs a great supply of glucose, a great supply of energy, which is why we have a high amount of mitochondria found in it. Cardiac muscle cells are more concentrated in the ventricles of the heart than in the atrium of the heart. The heart has 4 chambers, 2 atria, 2 ventricles. If you draw a cross, the ones above are the atria, the ones below are the ventricles. Why is this important? Or why is this significant? The ventricles are the part of the heart that are function, or the part of the heart that are involved or function to pump blood out of the heart. Whether the blood is going into systemic circulation or the blood is going into pulmonary circulation, it is going to be pumped out via the ventricles. A lot of force is going to be needed or it's going to be generated to perform this function, especially in the systemic circulation, which is why the ventricles will have more muscle fibers or muscle cells, so that they will have a stronger or a more potent excitation and a stronger contraction to produce larger amount of movement. In the atrium, there is also contraction, but the force of contraction in the atrium is in no way compared to the force of contraction in the ventricles. Thus, the significance, atrium, ventricle. Thus, the significance of the separation, or highlighting where muscle fibers are more concentrated in. Okay, now we're going to deal with the last muscle, smooth muscle. The cells of our smooth muscle, of this muscle type, the cells of smooth muscle are called fusiform cells. Let's do this one. So this is for our smooth muscle. It's called fusiform cells. And from this diagram, you can kind of tell why it's called fusiform cells. Because the cells, they fuse together. They're very, very closely and tightly packed. The cells of skeletal muscle fiber, which are the myocytes, and the cardiac muscle fiber, cardiomyocytes, they are also packed together, joined. They are not actually packed together per se. They are joined together by the connective tissue. But then in smooth muscle, they are more packed, like they are more tightly packed, more joined together, which is why they are given the name fusiform cells. Let's go down on our fusiform cells with the characteristics. First characteristic, shape. So our fusiform cells, it looks like a flame, if you want to draw a flame. But our fusiform cells, this shape is that it is elongated, and then the ends of our fusiform cells, they are tapering off, which means that centrally our fusiform cells, they are wide, they are fat. But when we are approaching the end, like after the central part, as it is coming together, it is tapering off, it is becoming smaller. That's what tapering off means. So elongated and tapering off. That's the shape. Then the control, similar to our cardiac muscle cell, similar to our cardiomyocytes. Smooth muscle cells, they are non-voluntary. They cannot be controlled by you and I, but then they are controlled by the autonomic branch of the central nervous system. So, autonomic and involuntary. You cannot control how your food is being digested, I guess. You can control the vasoconstriction of your blood vessels. Don't be scared, you have escaped. Location, the smooth muscle cell, that's the fusiform cells, they are majorly located in viscera and along the gastrointestinal tract, like the small intestine, the large intestine. That's where like fusiform cells principally associated with cells of the gastrointestine. Gastrointestine. So majorly gastrointestinal cells, GIT cells. Ain't writing all that. Striations are absent, as you can see. These are not striations. Don't be deceived. You know what? Let me just clean it up. There are no striations. There are no striations. This was just added defect too. There are no striations in this cell. It was added effect, I beg. There's no striation there. Oh, please. Oh. Who is this person I won't drew this? It's me. Who's this person who drew this diagram? So, nuclei. Nuclei or nucleus, it has just one nucleus per cell, and the nucleus is centrally located. Mononucleic, centrally located. Intercalated discs, no intercalated discs. There are no intercalated discs between the cells. Regeneration. In fusiform cells, regeneration is abundant. It's very much abundant because of how tightly packed they are. In this tight packing, like there are going to be some what we call myosatellite cells here, but we're going to be calling them myoblasts here, which those are the embryonic muscle cells. So there are a lot of myoblasts here, which enables smooth muscle to undergo regeneration properly. Regeneration, very possible, very abundant. So yes, not limited. Contraction threshold. Smooth muscle also follows the all or none law. Yes, it also follows the all or none law. And then connective tissue. It is also surrounded by connective tissue, endomysium, but in smooth muscle cell, we don't really have perimysium and epimysium. What goes on in smooth muscle cells is that, yes, we have endomysium, but even that endomysium is very, very small. So that means that, so what now happens, like how it is still able to stay tightly packed or closely packed together, it's not falling off or not getting destroyed, whatever, is apart from endomysium, the external lamina of the sarcolemma is attached to type 1 and type 3 collagen fibers, like just collagen fibers is attached to it. So that is what it has in place of perimysium and epimysium. It has endomysium and type 1 and type 3 collagen fibers holding it together. Great. In this video, we spoke about the muscle as an organ. We spoke about the functions or the properties of muscle, what makes the muscle an organ, and we delved deeper into the histology of muscle, the different muscle types, skeletal, cardiac, and smooth muscle. If you said any other thing, go and eat. You have successfully come to the end of this video. Please like this video. I hope you liked it, Sha. I don't want you to just press the button for pressing sake, but I hope you really liked the video, the explanations and the jokes in between. If you did, please like the video to encourage me so that I can be encouraged. Also subscribe so that you can get more notifications. When I post videos, you get notifications and you'll be able to watch those videos as my fan. And you can share this video to whoever needs it, preclinical students. If you know any 200 level, 300 level, 400 level, any paramedical student or medical student of any level, you can share this video to them. You can also share this video to an accountant or a lawyer. It is important to know what goes on in your body. Thank you for staying thus far. Have a great day and see you next time on Dicot Medical. Hey!